Welcome to this lecture on developmental biology. Today, we are going to explore one of the most fascinating phenomena in nature, embryonic induction and organizers. Have you ever wondered how a single, simple fertilized egg knows exactly how to build a complex organism with a brain, a heart, and limbs in all the right places? It all comes down to cellular communication. Let us dive in and uncover how cells talk to each other to build a life. To understand how this works, imagine building a massive, complex city entirely from scratch. You do not just have workers randomly placing bricks wherever they please. Instead, you have a foreman who shouts specific instructions to the workers nearby. The closer the workers are to the foreman, the louder the instructions they hear, prompting them to build a towering skyscraper. The further away they are, the quieter the instructions become, leading them to build a quiet residential park. In a developing embryo, cells act just like these construction workers, starting out completely naive. Now that we have our city-building analogy, let us look at the biological reality. In developmental biology, this process is formally known as embryonic induction. It is a highly coordinated interaction between two or more groups of cells. One group acts as the foreman, which we call the inducer. The inducer secretes chemical signals, known as morphogens, across the empty spaces between cells. The neighboring cells that receive these signals are called the responders. By receiving these chemical messages, the responding cells change their behavior and commit to a specific developmental fate. This brings us to an important question. Can any cell respond to these signals? The answer is no. For this communication to work, the responding cells must possess specific receptor proteins on their surface to catch the chemical signals. This critical property is known as competence. If a cell lacks the right receptors, it is entirely deaf to the inducer signal. It will not change its developmental path, no matter how much of the morphogen is present. Therefore, induction is a strict two-way dialogue, ensuring tissues form at the precise correct time and location. Before we move on to the mechanics, let us understand why embryonic induction matters so much to modern medicine. Without it, an embryo would simply grow into a disorganized ball of identical cells. Every major organ in your body formed because of these inductive signals. In regenerative medicine, if scientists want to grow a replacement liver or heart tissue, they must artificially replicate these exact signals to instruct raw stem cells. Furthermore, when these signals misfire or mutate during pregnancy, it can lead to severe birth defects or even trigger cancer, making this research life-saving. So, how do these chemical signals actually travel through the embryo? They move through a physical process called diffusion, which creates a concentration gradient. As the inducer secretes the morphogen, the molecules spread outward. This means that cells located very close to the inducer receive a massive dose of the chemical. Meanwhile, cells located further away receive a much smaller dose. This physical spread of molecules is the exact mechanism that allows a single group of organizing cells to send different instructions to different regions of the developing embryo based entirely on distance. This concept of diffusion leads us directly to the mathematics of morphogen gradients. We can model the concentration of an inducing molecule at any given distance using a steady-state diffusion and decay equation. Let us look at the formula. C of x represents the concentration of the morphogen signal at a specific distance, x, from the source. C0 is the maximum initial concentration, right at the inducer cells. The constant e is the base of the natural logarithm, describing continuous decay. Finally, lambda represents the decay length, which is how far the signal can travel before it is naturally destroyed. If distance increases, the concentration drops exponentially. If decay length increases, the signal travels much further. Now that we have our formula, let us see this with a concrete example. We are given an organizer tissue secreting a morphogen with an initial concentration of 100 units. The decay length is 20 micrometers. We need to find out if a cell located 20 micrometers away will become a muscle cell, which requires a threshold of 36.8 units. We substitute our known values into the exponential decay equation. The exponent simplifies to -1. Multiplying 100 by e to the power of -1 gives us exactly 36.8 units. Since this meets the exact threshold, the cell will successfully become a muscle cell. Let us apply this to another scenario involving the famous Spearman-Mangold organizer in a frog embryo. This organizer secretes a protein called chordin to induce the nervous system. In a healthy embryo, the initial concentration is 200 units, and the normal decay length is 40 micrometers. We want to find the concentration at a distance of 40 micrometers. We plug these values into our equation. The exponent again simplifies to -1. Calculating this out, we get approximately 73.6 units. This represents a strong, robust signal, which is more than enough to properly induce the formation of a normal brain and spinal cord in the healthy frog for the 4. But what happens if things go wrong? Let us compare the healthy embryo to a mutant frog embryo. In this mutant, a defective tissue environment causes the chordin protein to degrade twice as fast, cutting its decay length in half to just 20 micrometers. We use the exact same distance of 40 micrometers. Because the decay length is shorter, our exponent now becomes -2. The resulting concentration drops drastically to just 27.0 units. Biologically, this massive reduction means the cells fail to receive a strong enough message. This results in a severe birth defect where the frog lacks a properly formed nervous system. To visualize everything we have just calculated, let us look at this graph of a morphogen gradient. On the vertical axis, we have the morphogen concentration, starting high at the source. On the horizontal axis, we have the physical distance from the organizer. Notice the solid curve dropping exponentially as it moves to the right? This perfectly illustrates our decay equation. The horizontal dashed lines represent different biological thresholds. Cells close to the source cross the higher threshold and adopt a muscle fate. Cells further away only cross the lower threshold, adopting a skin fate. This diagram beautifully summarizes how physical distance dictates cellular destiny. We have covered a lot of ground today, so let us review our key takeaways. First, embryonic induction is the master communication system of the developing embryo, relying on inducers and competent responders. Second, these signals travel via diffusion, creating strict concentration gradients that we can model mathematically. Finally, the physical distance between the sender and receiver strictly dictates the instructions the cell receives, determining whether it becomes a brain, a muscle, or skin. You now have a solid understanding of how complex life forms from a single cell. Great job!